Hello everyone, am I audible? Can someone let me know if I'm audible? Hello, am I audible? Anybody? Reshma, Meghna? Okay, please turn on your mic and let me know because I won't be looking at the chat box after I go into presentation mode. Okay, so turn on your mics and speak up. This is an interactive session, so since there are a lot of questions in the past couple of weeks, few questions, direct questions, we'll discuss it. Okay. Is my presentation visible? Can someone please answer? Is my presentation visible? I am in presentation mode. Please turn on your mic and answer me. Is anybody there? Can you please answer? Is my presentation visible, guys? Okay. Let me just reconnect. Okay. So this is week 10 live session for experimental biotechnology. Since there are no participants as of now, I will just start the recording of the session. Okay. So the first topic is about the nucleotides and phosphate sugar and the base pairs of DNA molecule, that is the basic structure, the parts of a nucleotide. A DNA molecule is just a second. Yeah. So a DNA molecule is nothing but deoxyribonucleic acid and is made up of repeating units of nucleotides. So nucleotides each consists of three main parts. One is the sugar backbone, a phosphate group attached to the sugar moiety. and a a purine or a pyrimidine base. So a 5 carbon sugar, that is the deoxyribose sugar. It is a pentose sugar, meaning it has 5 carbons, and the sugar is deoxyribose, which is slightly different from the sugar in RNA, which is ribose. So you can see this has a deoxyribose instead of a complete ribose sugar because it lacks the oxygen atom at 2 prime carbon position. So this is the main oxygen in the ring. This is the first carbon, second carbon, third carbon, four, fourth carbon, and the fifth carbon. So in the second moiety, it does not have an oxygen atom. So it is a deoxyribose. It also has a nitrogen base, nitrogen containing molecule that store genetic information. These are the bases which define the sequence, and there are four nitrogenous bases: adenine, guanine, cytosine, thymine. So adenine and guanine are purines, and cytosine and thymine are pyrimidines. Adenine and guanine are double ring structures. You can see here there are two rings, whereas cytosine and thymine are single ring structures. The nitrogenous base attaches to the first carbon of the deoxyribose. This is the first carbon, and the nitrogenous base is attached to the first carbon of the carbon ring. These base pairs, specifically A with T and G with C. So A pairs with T and G pairs with C. This is their usual complementarity via hydrogen bonds to form rings of the DNA double helix. Then it also has a phosphate group. A phosphate group is attached to the fifth carbon of the sugar, and it plays a crucial structural role by linking the 3' carbon of one nucleotide's sugar to the 5' carbon of the next nucleotide's sugar. You can see here, this is the 3' of one, this is the nucleotide base, and this is the 3' of one sugar, and this is the 5' of the other complementary sugar. This linkage forms the sugar phosphate backbone, which gives the DNA strand stability and directionality. That is 5' to 3'. So DNA sequence is generally written from 5' to 3'. Then each nucleotide looks like a phosphate group that is attached to a sugar, that is deoxyribose in case of DNA, and then attached to a nitrogenous base. Nucleotides join together by phosphodiester bonds between the phosphate group of one nucleotide and the sugar of the other nucleotide. So, the bond between the phosphate group of one nucleotide and the sugar, that is the third carbon of the other nucleotide, you have the OH group. So the phosphate and the OH are bound together by a phosphodiester bond. Long chains of nucleotides form a DNA strand, and two strands twist into the famous double helix. So this is usually how the sequence is bound. This is one strand and this is the other strand. You have the sugar, nitrogenous base, etc., etc., and then you have the sugar of the bottom uh, nucleotide and the sugar of the top nucleotide, and these two are bound to each other by the phosphodiester bond, and then this strand and this strand are bound to each other by the hydrogen bonds between the nucleotides. So this is one single strand of DNA and this is the other single strand of DNA. So these two are complementary strands. So you can see you have a continuation like this of nucleotide attached together using phosphodiester bonds. So once attached like this in a sequence, this single strand and this complementary single strand will be bound with the hydrogen bonds. And when two chains like this are attached with multiple hydrogen bonds down the line, they will eventually twist together to form a double helix in this way. So this is one strand and this is the complementary strand to it. In between we have the nucleo nitrogenous bases which form the complementary hydrogen bonds. DNA is built from four different nitrogen bases. These bases are classified into two groups. Like I said, purines and pyrimidines. So the purines are double ringed structures, and purines are larger molecules made up of two fused rings. You can see this is the major ring and this is another ring. And these rings are made up of carbon and nitrogen atoms. The two purines found in DNA are adenine and guanine. Their main features are they have double ring structure, a six-membered ring fused with a five-membered ring. Because of their size, purines always pair with pyrimidines, which are smaller and have a single ring, to maintain the uniform width of the double helix. Then we also have the pyrimidines, which are single ring structures. You can see each of the pyrimidines here are single ring. In DNA, we have only cytosine and thymine. In RNA, instead of thymine, we have uracil. Then pyrimidines are smaller molecules with a single ring. In DNA, pyrimidines are cytosine and thymine. And base pairing in DNA is when each purine pairs with a pyrimidine through hydrogen bonds. This is crucial to maintain the DNA's double helical structure. So adenine, which is a purine, pairs with thymine, which is a pyrimidine. So we have AT bond, which is a two hydrogen bond. And then we have guanine, which is a purine, and cytosine, which is a pyrimidine. And then we have a GC bond, which is a three hydrogen bond. This pairing rule is also known as Chargaff's base pairing rule, ensures the DNA molecule maintains a consistent structure and diameter throughout the length. So basically DNA contains two purines A and G. These bases are double ring structures and pairs with single pyrimidine rings T and C to form a stable DNA double helix. So this was about the DNA and their base pairs and the basic structure of it. So next comes about the tethering proteins. So during DNA replication, several proteins work together to ensure the accurate and efficient synthesis of new DNA strands. One important group of proteins involved are tethering proteins. So, tethering basically means to clamp together. So, it is also known as sliding clamps or clamp proteins. So, what are tethering proteins? Tethering proteins are ring shaped proteins. Example, beta clamp in prokaryotes and pCNA in eukaryotes. So, pCNA is proliferating cell nuclear antigen, which is a clamp protein. Then, their main job is to bind to the DNA polymerase and hold it firmly onto the DNA template during the replication process. So, the function in DNA replication is DNA polymerase is the enzyme that is responsible for adding nucleotides to the growing DNA strand. This tends to fall off the template after adding few nucleotides if it is not stabilized. So the tethering protein clamps or basically helps the DNA polymerase hold on to the uh, DNA strand, significantly increasing its processivity, the number of nucleotides it can add without dissociating. Then this ensures rapid and continuous DNA synthesis, high fidelity, which is the accuracy in replication, and efficient progression of the replication fork. So this is the major function of tethering proteins. So this is a table in which you can see different proteins which attach in this process. We have the helicase, which unwinds the double helix of DNA so that the DNA pol can access the single stranded DNA molecule, and we have single stranded binding proteins or the SSB proteins. This prevents the single strand DNA from re-annealing to the complementary strand. So once helicase opens the strands, there is very high chance of the strands to re-anneal together. So if it re-anneals, the DNA pol cannot bind to the single strand. So to prevent this re-annealing, we have single stranded proteins binding here. So this is how a replication fork looks. We have the helicase which has unwound the DNA. You can see the double helix has become a single stranded here. Then we have DNA pol. So DNA pol on the leading strand, and when we have DNA pol on the lagging strands. So in the DNA, in the leading strand, we have continuous synthesis of the new strand. In the lagging strand, we have Okazaki fragments, which will be later joined to each other. Then we have single stranded proteins bound onto the single stranded DNA, and we have parental DNA helix here, which is bound with topoisomerases, which uncoils it, and then we have newly synthesized strands here. We have RNA primer for the Okazaki fragments, and we have the clamp loader here. So you can see it helps in holding on to the DNA strand so that the DNA pol doesn't dissociate. So next is the purines and pyrimidines in a DNA molecule are linked through what type of bond? So we have already discussed this. It is hydrogen bonds. So you can see that the purines and the pyrimidines are attached to each other by hydrogen bonds. Adenine binds to thymine in case of DNA and uracil in case of RNA. In the same way, guanine binds to cytosine in case of both DNA and RNA. And adenine and thymine have two hydrogen bonds. So each of them has one donor and one acceptor for this bond. And cytosine and guanine has three hydrogen bonds, where cytosine gives one donor and has two acceptors, and guanine has two donors and has one acceptor. So this is the hydrogen bonds between the purines and pyrimidines. Next is calculation of melting temperature. So how to calculate the melting temperature? So they have also given a hint here, that is we have to take in the consideration of the primer length. So as we can see, the primer length exceeds 14 base pairs. So there are two ways you can calculate melting temperature of primers. So if the base pair is less than 14, you can directly go with the usual melting temperature formula, which is 4 degrees into G plus C and 2 degrees into A plus T. This should give you an approximate value if the primers are small. But if the primers are longer in length, then we need to have more accuracy. So we have the 64.9 degrees centigrade, that is the melting temperature, where you calculate it using 41 degrees centigrade into G plus C minus 16.4 divided by number of nucleotides. So we should use a length aware melting formula. So the formula is this: the 64.9 plus 41 plus G plus C minus 16.4 into N, where N is the primer length. So forward primer in our case is this, and this is the reverse primer. Both of them have 24 nucleotides in them, or 24. Uh, the length of each is 24. So we have to just substitute the number of G's and C's in this formula. So in this case, we have 12 G's, 12 G plus C, and in this case, we have 10 G plus C. So here we have 8 G's and 4 C's, which is 12. So after substituting into the formula, we get the answer around 57.1. Then in the reverse primer, we have 4 Gs and 6 Cs, and together they give 10. So after substituting it into the formula, we get the answer 53.5. So approximately by rounding off, we have an answer of 57 and 53.4. So if you don't use all the decimals during the calculation, if you use the first decimal or first two decimals, you will get a rounded off value of 57 for forward and 53.5 for reverse. Next is Taq DNA polymerase. So, Taq DNA polymerase is a heat stable enzyme which is isolated from a thermophilic bacterium, Thermus aquaticus. This usually is present in the hot springs, and due to the higher temperature, its DNA pol has the stability to function at high temperatures. Because of this origin, the enzyme works best at high temperatures. So during PCR, we know our different steps: the denaturation, annealing, and extension. So our denaturation, which completely separates the DNA bands, is around 94 to 95 degrees. So at this temperature, our usual DNA pol doesn't work. So that's why we have Taq DNA pol, which works best at 72 degrees and is also stable at 94 to 95 degrees. So during PCR, the extension or the elongation step, where the DNA Taq pol adds nucleotides to the growing DNA strand, is carried out at an optimal temperature of 72 degrees. So that's why we have an extension of 72 degrees. This temperature ensures maximal activity of the enzyme and high fidelity DNA synthesis, so it maintains the accuracy. Thus, the optimum temperature is 72, and how a typical PCR temperature relates to each step is: denaturation is carried out around 95 to 94 degrees centigrade, where the DNA strands separate. Annealing is carried out around 50 to 65 degrees, depending on the primers, enabling the primers to bind to the template. And then we have extension, where the new nucleotides are to be added by the Taq pol. So the Taq pol synthesizes new DNA at 72 degrees centigrade. This is the basic temperature guide. Next is a DNA sample of 1,000 base pairs was subjected to PCR under given conditions. The forward and the reverse primers were of equal lengths, that is 23 base pairs each, having a TM value of 64 and 62 degrees respectively. The PCR conditions were as follows: initiation is 95 degrees for 5 minutes, denaturation is 95 for 45 seconds, annealing is 65 for 45 seconds, elongation is 72 degrees centigrades for 120 seconds. Then final extension, we have 72 degrees Celsius for 10 minutes. So this is the program setup in the PCR, but the PCR resulted in a low yield product. So what could have been the cause of the low yield is the denaturation, the duration of denaturation is less, or the duration of annealing is less, or the annealing temperature is high, or the TM values are high. So in PCR, annealing is crucial step. This is when primers bind to or anneal to their complementary sequences. The annealing temperature TA must be carefully optimized. It is usually 3 to 4 degrees Celsius below the primer melting temperature to ensure efficient binding. So, in the given conditions, forward primer TM is 64, reverse primer TM is 62. So, an average TM for both these is 63. So, the ideal temperature should be TM minus 3 to 5 degrees centigrade, which means it should be between 58 to 60 degrees centigrade. But in our case, the annealing temperature is 65 degrees centigrade, which is higher than both the primers melting temperature. So it will not help in the proper binding of the primer to the strands. So at 65 temperatures, the temperature is slightly higher than both primer TMs, which causes poor or incomplete primer annealing, reduced binding efficiency, low yield of the amplified product. So that is the reason the PCR resulted in a low yield of product, because the annealing temperature is high. Next is the role of SSB proteins. So, like I said, SSB proteins are single stranded binding proteins. These are essential for these are essential accessory proteins which are involved in prokaryotic replication and eukaryotic replication. So, in case of prokaryotes, their equivalents are RPA replication protein A, RPA like replication protein A in eukaryotes, and they have their major role to help the to help prevent the re-annealing of the single strands together. So they do not catalyze any reactions themselves, but they play a crucial structural and protective role during the replication process. So during the DNA replication, excuse me, during the DNA replication, the enzyme helicase unwinds the parental double-stranded DNA helix. So then we get two single strands to serve as templates for the synthesis of new strands of DNA by DNA poly. However, the single strand is unstable and prone to several problems. So not only they can re-anneal, but they can also get degraded, or they can also, have damages in their sequence or anything, because single strand is not very stable when compared to the double strand. And there can be re-annealing, or it can form secondary structures, or it can get degraded. So the separated strands can easily form base pairs again and resip into a double helix. So this will not allow the DNA pol to bind and replicate. So it will prevent replication machinery from accessing them. Other than this, it can also fold back on itself if it has complementary sequence within its own strand. Also, it can complement to each. It can complement upon itself, forming hairpins or secondary structures which can block replication. Then degradation. So single stranded DNA is more susceptible to degradation by nucleases. So all these things will be prevented by the binding of single stranded proteins. So, single stranded proteins bind tightly and cooperatively to the single stranded DNA to solve the above problems that were mentioned. And the main functions are preventing re-annealing. The single strand binding proteins coats the single strand and keeps them apart physically, blocking the base pairing between the complementary strands. It ensures that the template strand remains single stranded and available for DNA polymerase. It also prevents the secondary structure formation by binding along the length of single strand DNA. SSBs prevents it from folding into hairpins or other structures that would hinder the replication process. Other than this, it protects by shielding the single strand regions from enzymatic degradation, and it also stabilizes the replication fork in this process and helps recruit other proteins other than DNA pol. It also helps recruit the primase, maintaining an open accessible template. So in summary, the single strand binding proteins coat the single stranded DNA during replication to prevent re-annealing, secondary structure formation, degradation, and ensuring that the template remains accessible to the replication machinery. Next is that we have a forward sequence of primer, then we have a reverse sequence of primer. The given set of primers did not yield any product for PCR. So, what could be the possible reasons the primers do not work? So, it could be because of the complementarity of the primers, or could it be because of the length of the primer, melting point, too high or too low? So, we can see that the length of primer is sufficient enough. It has uh, 1, 2, 3, 4, 5, 6, 7, 8, 9. It has around 18 to 19 base pairs, so the length is sufficient. And the melting temperature, it has like proper GC content in it, around 60%. So this should not give a very high or low melting point. And since they have not mentioned any PCR programs, the other option is to check the complementarity. So we can see that the 5 prime and the 3 prime forward and reverse primers are exactly complementary to each other. So the G is complementary, CG, GC, again we have AT, then we have GC, AT, TA, AT, So throughout the length, every uh, nucleotide is complementary to the other nucleotide from the forward to the reverse. So instead of binding to the template DNA, these bind to each other more efficiently because they are smaller in size and they can exactly bind to each other before as soon as you add it into the reaction mixture. So only after you start your PCR step, your template will get separated, or your after your annealing, your um, primer can bind to the DNA. But as soon as you add it into the reaction mixture, because they are completely complementary to each other, the forward and reverse primers bind to each other. So the forward and reverse primers are perfect complements of each other. This means they can bind to each other instead of the template, forming primer dimers. So this is a major problem. We have to be sure that primer dimers are avoided so that we get our amplified product. Even if the primers are very correctly designed, if they are complementary to each other, we will not get any amplified product. So a primer dimer will prevent amplification because the primers are used up binding to each other instead of the target DNA. So if the primer doesn't bind to the target DNA, there cannot be any synthesis of the amplicon, or there cannot be any extension. So next is about the tacpol. So the tag DNA pol is thermostable DNA polymerase, originally isolated from Thermus aquaticus. It is a workhorse of PCR because it can withstand the high temperatures as required for the DNA denaturation step initially, and its enzymatic activities are what determines both its strengths and its limitations. Though it is functional at a high temperature, it also has certain limitation. So Pol activity 5 prime to 3 prime. TAC can synthesize DNA by adding nucleotides to the 3 prime end of the primer, complementary to a DNA template. So this is the essential activity required for PCR. So TAC polymerase does have this activity, and 3 prime to 5 prime exonuclease activity is required for proofreading. So some polymerases like PFU have 3 prime to 5 prime exonuclease activity that removes incorrectly incorporated nucleotides, which is called proofreading. TAC polymerase lacks this activity, so it cannot proofread the newly synthesized strand. The consequences: TAC makes more errors during DNA synthesis, that is approximately one error per 9,000 to 30,000 bases, because it cannot correct its mistake. So in every replication by uh, like different polymerases, there is possibility of errors and mistakes, but the rate at which it can make mistakes is what defines its fidelity. So even though TAC is functional at higher temperature, because it lacks the exonuclease activity, which is helpful in proofreading, its error rate is much higher. So 5 prime to 3 prime exonuclease activity. This activity removes the nucleotides ahead of the polymerase used in DNA repair or nick translation. TAC polymerase has very limited or negligible 5 prime to 3 prime, and it is not important during replication, it is for repair. So this is anyways not uh, an important characteristic for PCR, but it still has a very little exonuclease 5 prime to 3 prime activity, but it does not have 3 prime to 5 primase exonuclease activity, which is really important during replication process to avoid mistakes. It has thermostability, that is the reason we use it in PCR. So TAC polymerase can withstand repeated heating cycles, and it is stable at high temperatures. So this is a table of its activity that it has. And most commonly emphasized is the lack of activity of 3' to 5' proofreading, which explains why PCR products made with TAC may have occasional point mutations due to error in the replication process, which is not corrected. Next is PFU DNA polymerase is isolated from which bacteria? So, as the name suggests itself, P stands for Pyrococcus, FU stands for Furiosa. So, PFU DNA pol is thermostable DNA pol used in PCR when high fidelity is needed, because it has 3' to 5' proofreading activity, which is absent in the TAC pol. So it when you're synthesizing a new sequence for, maybe transfection or any kind of knockdown studies or over expression studies, you need to use DNA pol which can proofread, because you don't want to incorporate mutations in your newly synthesized strand. If your aim is to only amplify the product and check the expression, then you can easily use TACPOL, because you will not further process your amplicon that is being amplified in the process. But when you have to use your amplicon for downstream experiments, you need to make sure there are no mutations incorporated. So PFU is isolated from hypothermophilic archeon, uh, Pyrococcus furiosa. Sorry, it's not a bacteria, it's an archeon. So Pyrococcus furiosus lives in extremely hot environments, that is uh, almost 100 degrees centigrade, which is why its polymerase is stable at such high temperatures. PFU polymerase is slower than TAC but much more accurate due to its proofreading ability. So every pol has its own plus and minus. So depending on your experiment, you can choose whether you need to use TAC or PFU. Next is a problem where X is the number of double helical strands present in a sample. How many number of double helical strands will be present after 10 cycles in a PCR machine? So X is taken as 2. So it's usually 10 power the number of cycles, or sorry, it's usually the X 2 into the number of cycles is the exponential um, increase. But because they are asking about the double helical strands here, it is double of that. So PCR is an exponential process, so after each cycle the number of double helical strand doubles. So initially given dsDNA molecule is 2, so X is the number of double helical strands. So 2 double helical strands are present and the number of PCR cycles are 10. So the formula is simple. Number of double helical strands after n number of cycles is X into 2 to the power of the number of cycles, where n is the number of cycles. So the number of cycles that were given were 10. So 2 into 2 power 10, which is 2448. So that is your answer here. Next is Chlamydia trachomatis. It is a bacterial pathogen that primarily infects the cervix but can ascend to also the upper reproductive tract, causing pelvic inflammatory disease. So, pelvic inflammatory disease is the main mechanism which leads to female infertility, and asymptomatic it is an asymptomatic infection. Most chlamydia infections in women are asymptomatic, so they often go untreated. This allows the infection to persist and further damage the reproductive tract over time. Ascending information. So chlamydia can move from cervix to the uterus to the fallopian tubes and the ovaries. So the inflammation and scarring, that is immune response to the infection, causes inflammation in the fallopian tubes, and repeated chronic inflammation leads to fibrosis and scarring of the tissue, narrowing or blocking the tubes. Consequently, tubal blockage prevents the ovum from reaching the sperm and causing infertility issues. Even if conception occurs, scarring increases the risk of ectopic pregnancy, that is, implantation into the fallopian tube itself instead of the uterus. So the major points to remember is chlamydia is the most common bacterial cause of female infertility worldwide, and also Neisseria gonorrhoeae can also cause PID, but its infection is more acute and symptomatic, leading to early treatment. So chlamydia is a bigger hidden threat. HIV and hepatitis B do not directly affect the fertility, though HIV can indirectly influence the reproductive system health, overall health through general deterioration. So PID can be caused by different types of bacteria. About 85% of the PID occurs by sexually transmitted infections such as chlamydia, so and gonorrhea and also Mycoplasma genitalium. Douching can move the bacteria found in the vagina to other reproductive organs, and other causes can be a ruptured appendix, a bowel infection, vaginal infections, surgical procedures such as dilation or curettage, and intrauterine devices, that is IUDs. So these are all the causes of pelvic inflammatory disease, and the major cause is chlamydia trachomatis. Next is, which of the following is not a method to detect individual human beings based on their genetic differences? We have fingerprinting using variable number tandem repeats, fingerprinting using short tandem repeats, mitochondrial DNA analysis, 16S and 23S rRNA sequencing. So, fingerprinting using variable tandem repeats are repetitive sequences in nuclear DNA that vary between individuals to individuals. It can be used for human identification in forensics and paternity tests. Short tandem repeats are short repeats, repetitive sequences, highly polymorphic between individuals, also widely used for DNA profiling and forensic analysis. Mitochondrial DNA analysis is inherited maternally and shows some variation between individuals. Useful for identifying individuals, especially when nuclear DNA is degraded, or we have old samples or hair or etc. etc. But however, 16S and 23S RNA genes are highly conserved sequences and not used for human identification. It is used for bacterial taxonomy and identification. Next is a DNA sample of 2000 base pairs was subjected to PCR under the given conditions. So the conditions are initiation 95 degrees for 5 minutes, denaturation 95 degrees for 45 seconds, annealing 55 degrees for 45 seconds, elongation 72 degrees for 120 seconds, and final extension 72 degrees for 10 minutes. So how much time does it take for 35 cycle run? We have to calculate. So usually the repeats will be for the cycle that includes the denaturation step and annealing and elongation. So the first we can convert everything into the same units, so it will be easier. So we can convert all the seconds to minutes. So 45 seconds is 0.75 minutes, 45 seconds, and 120 seconds is 2 minutes. So the total time per cycle. So these three steps are only involved in the repetitive cycles. So the total time per cycle is 0.75 plus 0.75 plus 2, which is 3.5 minutes per cycle. So for 35 cycles, we just multiply it by 35 and we get 122.5 minutes. Next, we have the initiation, which is only done once, and final extension, which is also done only once. So that is 5 minutes and 5 minutes. So 122.5 plus 5 plus 10 gives you 137.5 minutes. So the total program will take 137.5 minutes to complete 35 cycles. Next is GC content. So what is GC content? GC content is the percentage of guanine and cytosine nucleotides in a DNA sequence. So it is basically calculated by total number of G plus C by total number of bases into 100. So, why GC content matters in PCR? The primer template binding stability, that is GC base pairs, form three hydrogen bonds, whereas AT pairs only two hydrogen bonds. Higher GC content has stronger binding and therefore will lead to higher melting point. TM depends on GC content. Primers with very low GC may not efficiently bind to the annealing temperature. Primers with high GC form strong secondary structures or hairpins or primer dimers that reduce the efficiency. So we need to have optimal percentage of GC. So even during annealing, ideal primers should have similar TM values and usually 2 to 3 degrees within the range. The difference should be only 2 to 3 degrees between each other to ensure both forward and reverse primers bind or anneal properly. So, extreme GC content can disrupt this balance. So basically, these are the standard features for primers. The primer length is usually 18 to 25 bases. GC content should be 40 to 60%. Melting temperature should be between 52 to 65 degrees. GC clamp, that is the last one or two bases at the 3 prime end. This is optional because at the end, if you have a GC clamp, it helps in stable binding. If you have 80, sometimes it might dissociate. So GC content of approximately 50% is considered optimal. It should not exceed 60 to avoid secondary structures, and it should not be less than 40, which will lead to weak annealing. Both of these will lead to inefficient PCR. Other tips are you can include a GC clamp at the 3 prime end to improve the priming and avoid long stretches of GC more than the four bases of GC continuously because it can again bind to each other and form hairpin structures. So check for complementarity between primers to prevent primer dimer formation. So these are the basic tips for primer designing, uh, which will help in efficient PCR product. Next is regarding the magnesium ions. So the magnesium ions are crucial for the Taq polymerase to function because they are cofactors and they stabilize the interaction between DNTPs and the DNA template. It is also important for enzyme activity, fidelity, and primer annealing. Typical optimal concentration, that is for standard PCR with Taq polymerase, MgCl2 is usually in the range of 1.5 to 2 millimolar. Too low magnesium can decrease the enzyme activity and lead to low yield. Too high magnesium can cause non-specific amplification and misprime the primers and leading to random amplicons. So this is the table. 1.5 to 2 micromolar is too low because micromolar concentration is very less than needed. 1.5 to 2 millimolar is optimal. 1.5 to 2 nanomolar is way too low. And 1.5 to 2 molar is very high and would precipitate the DNA. Next is about the different steps in PCR. The process of binding of primer to single stranded DNA, like we discussed, is annealing. So, denaturation is double stranded DNA is heated to 95 degrees to separate into single strands. Annealing is temperature is lower to 50 to 65 so that primers bind efficiently to their complementary sequence on the single stranded DNA. Elongation is where the Taq polymerase synthesizes new DNA strands starting from the primers, and initiation is generally when you start the DNA synthesis and not primer binding. Next is about Carrie Mullis. So, Carrie Mullis was an American biochemist who developed PCR in 1983 while working at a California biotech company. He realized that short DNA sequences, that is primers, can be used to target specific regions of DNA, and with repeated heating and cooling cycles, these regions can be amplified exponentially in vitro. So the recognition and Nobel Prize was given, in 1993 in chemistry for the invention of PCR. PCR has since become a long foundation technique in molecular biology used in DNA cloning, forensics, medical diagnostics, genetic research, and sequencing. So you can see since 1983 how the PCR has also improved and developed, and currently this is how our PCR instrument looks. Originally, Taq polymerase wasn't used. A DNA pol was used from E. coli, and because it degrades, it was added fresh for each cycle. So it becomes tedious to continue it again and again, adding freshly for many cycles. So later, Taq pol was used because it is thermostable and allowed automation of the cycling, making it more practical without tedious addition at every cycle. So this is Dr. Carey Mullis, and this is what he invented. And so it is also said that science consistently produces a new crop of miraculous truths and dazzling devices every year. As you can see, the same device also has been evolved so well. So many inventions, such way has led us to carry out our research at a very advanced level. Next is the assertion and reason. The annealing temperature is always kept higher than the primer melting temperature in a PCR method. This is the assertion, and the reason is at primer melting temperature, the two strands are completely dissociated from each other. So A is false, definitely, because annealing temperature is not kept higher, but it's kept little lower than the primer melting temperature, as we discussed in earlier topics also. If the primer melting temperature is, for example, 63, the annealing temperature should be 3 to 5 degrees less than that. That is, it should be between 58 to 60. Then, so A is definitely false, so it eliminates all the first three options. R is actually partially true. It depends on what sense the statement is made. If it means that its main function is to dissociate the strands from each other completely, then yes, primer melting temperature does dissociate it, but it is not throughout, because melting temperature itself is defined as 50% of the DNA strand will be dissociated. So half the population will be dissociated, half of the population will be dissociated, half will be still bound. So in terms of dissociating the two strands efficiently from each other, it is correct, but it's not throughout. Okay, so the only option left is this. So A is false and R is true. So I guess we are done with all the questions. And I guess since there are no students this time, okay, the meeting also has been logged out because there were no attendees. So I will just upload the recording so that everybody can access it. Yeah, so the meeting is closed because there were no attendees this time. So the both the presentation and the video will be accessible. The presentation will be uploaded in the drive and the video will be uploaded on the YouTube. You can access the link of the video. If you have any doubts regarding this session, you can always mail me to this email ID. I guess I can close the session now. Okay. Thank you, everybody.